Hey everyone, and welcome to this explainer. I was thinking the other day, have you ever wondered if our bodies come with a sort of factory default setting? Think about it like a brand new smartphone, straight out of the box. It has all these standard settings that dictate exactly how it interacts with the world way before you start tweaking things or downloading apps. Well, it turns out biology absolutely has its own version of a factory default. Today, we're exploring the fascinating world of genetics to understand the exact benchmark scientists use to map out life itself. We're going to decode the secret language of your DNA, look at how populations actually shift over time, and discover how this fundamental biology is literally revolutionizing modern medicine. Okay, let's dive right in. Our agenda today covers four main areas. 1. The genetic baseline. 2. Wild type versus mutant. 3. The shifting standard. And 4. Medicine and your genes.
So, starting with section 1, the genetic baseline, setting the standard.
I want to kick things off by asking you a question. What does the word normal actually mean in genetics? I mean, we throw that word around all the time in our daily lives, but in science, normal needs to be measurable. It has to be incredibly precise. It isn't a judgment. It's a specific, hard biological data point. So how do scientists actually pin down what the normal version of a trait is when there's just so much diversity out there in the natural world?
Well, the answer lies in this absolutely fundamental concept, the wild type allele. Basically, this is the standard, most prevalent version of a gene found in a natural wild population. It serves as biology's ultimate baseline reference point. So when geneticists are studying a species, they use this wild type as the benchmark to compare against any altered or mutated versions of a gene. And crucially, this wild type allele typically produces a functional protein. It's that specific functional protein that's responsible for creating the standard physical traits, what scientists call phenotypes, that an organism actually displays out in nature.
At the end of the day, a wild type is really defined by the math of the crowd. By definition, for an allele to earn that wild type title, it literally must be observed in the majority of a natural population. It just needs to cross that 50% threshold. So if 51% of a population out in the wild carries a specific version of a gene, boom, that version represents the baseline. It's the prevailing genetic reality for that group. Now, as you can imagine, geneticists are dealing with massive, massive amounts of data, right? So they use a specific genetic shorthand to keep everything organized. When you're looking at genetic notation, you always want to look for the plus sign. It's usually a superscript, like a lowercase a with a tiny plus sign right next to it, and that symbol designates the wild type default. It's actually a pretty elegant system. If you just see a standard uppercase or lowercase letter, that typically tells you about the dominant or recessive nature of a variation. But the second you add that plus sign, you are officially looking at the genetic baseline.
Moving right along to section 2, wild type versus mutant, deviating from the norm. Now, I know when we hear the word mutant, pop culture completely trains us to think of like sci-fi monsters or superheroes. But in genetics, the reality is way more common and honestly, much less dramatic. While wild-type alleles represent the norm, a mutant allele is simply a variation caused by random DNA changes. That's really all it is, just a deviation from the baseline. However, these seemingly simple, random little DNA changes can result in entirely different physical characteristics for the organism.
To really visualize how this works, let's look at one of the most famous subjects in all of genetic research, Drosophila, better known as fruit flies. In the natural population of these flies, the wild-type allele dictates that they have bright red eyes. That's their factory default. But a simple, random DNA change, a mutant allele, completely alters that physical characteristic, giving you a fly with white eyes. No laser beams, no superpowers, right? The white eyes aren't a mistake. They are just the mutant counterpart to the bright red wild-type standard.
We can also see this perfectly illustrated in Gregor Mendel's foundational studies with pea plants. If you took high school biology, you definitely remember these plants. In Mendel's fields, the wild-type allele, that standard baseline, coded for plants with tall stems and nice round seeds. The variations he observed, things like dwarfism or wrinkled seeds, well, those were the direct result of mutant alleles. By contrasting these two, the standard tall and round versus the mutant short and wrinkled, Mendel essentially helped unlock our entire understanding of heredity.
All right, section 3, the shifting standard, a dynamic baseline. Here is where we hit a major biological plot twist. The truth is, wild types are not fixed forever. Actually, scratch the idea of a rigid code locked in a vault, because nature is incredibly dynamic. Because species cover such huge geographic ranges, what's considered a wild type can actually vary slightly between different isolated populations. Plus, biology is so resourceful that you can have multiple, entirely different DNA variations that surprisingly produce the exact same physical trait. So, how does a baseline actually change across a population over time? It usually follows a pretty fascinating path, heavily influenced by environmental shifts. Step 1, you've got a stable environment where the current wild type dominates perfectly. Everything is humming along. But then, step 2, a big environmental pressure or shift happens. Maybe the climate completely changes or a brand new predator shows up. Step 3, suddenly, the traits produced by the old wild type are a huge disadvantage. But wait, a random mutant allele happens to be perfectly suited for this new environment, allowing those mutants to survive and thrive. And finally, step 4, through this evolutionary shift or genetic drift, that once rare mutant allele spreads like wildfire. It crosses that 50% threshold we talked about earlier, effectively overthrowing the old standard and becoming the brand new wild type. The mutant literally becomes the master.
Which brings us to our final part, Section 4, Medicine and Your Genes, Personalized Health. Now, understanding wild-type alleles isn't just for studying fruit flies or pea plants. It is absolutely crucial for human health right now. It's the cornerstone of a cutting-edge field called pharmacogenetics. Pharmacogenetics is all about predicting exactly how your specific body will metabolize prescription medications. And how do doctors figure that out? By identifying your wild-type alleles. By looking at your genes to see if you have the standard baseline alleles for specific enzymes, doctors can finally move away from that old one-size-fits-all medicine and start completely personalizing treatments to fit your exact genetic code. To understand just how vital this is for medical treatments, we have to look at the CYP450 enzyme. You can really think of CYP450 as the main chemical processing plant of human medicine. It's a whole family of enzymes responsible for metabolizing a vast majority of the prescription medications we take every day. Whether your body processes a drug efficiently, way too slowly, or dangerously fast depends entirely on the genetic instructions coding for these specific enzymes. And this brilliantly illustrates exactly how enzyme variations influence a patient's drug response. Look at it this way. If a patient has the wild-type CYP450, their factory default is perfectly intact. Their body will exhibit standard drug processing and baseline metabolism, meaning a standard dose of medicine from the pharmacy will work exactly as intended. However, if a patient carries a mutant CYP450 allele, the instructions for their internal chemical processing plant are changed. This leads to altered drug processing. They might clear the drug from their system so fast that it doesn't even work, or worse, it might build up in their bloodstream, leading to serious potential adverse reactions. Knowing this genetic baseline isn't just trivia. It's literally a matter of life and death in modern medicine.
So I want to leave you with a thought to chew on. The next time you take a simple medication, ask yourself, are your enzymes wild type, efficiently running that standard factory default? Or is your body playing by its own completely unique, mutant genetic rules? Understanding the genetic baseline isn't just about figuring out the natural world around us. It's the ultimate key to understanding ourselves, unlocking personalized healthcare, and embracing the incredible variations that make life so incredibly dynamic. Thanks so much for joining me on this explainer today. Keep asking questions and keep exploring.